So today we are going to discuss about the process of formation of urine. Okay. Now there are three steps which are involved in the formation of urine. So the first step is known as ultrafiltration. Okay. Second step is known as selective reabsorption, and the third step is known as tubular secretion. Okay, so there are three steps are involved. The first is called as ultrafiltration, or which is also known as glomerular filtration. Second step is known as selective reabsorption or tubular reabsorption, another name. Okay, then last is called as tubular secretion, or it is also called as augmentation. Okay, so these three steps which are a part of process of urine formation. So we are going to discuss the first part, that is ultrafiltration. Okay, if you see that this is a structure of Malpighian body. Okay, so you can remember this. This is a part of a renal artery or branch of the renal artery called as afferent arteriole. So this is afferent arteriole. Now that afferent arteriole, this is the afferent arteriole. It is going to enter into the cup-like structure. So can you see this is a cup-like structure? That cup-like structure is called as Bowman's capsule. I hope you can remember. That Bowman capsule has outer layer is called as parietal layer, inner layer is called as visceral layer. That visceral layer, it is in contact with the glomerulus. Okay, it is in contact with the glomerulus. So what is glomerulus? You can see this is called as afferent arteriole. That afferent arteriole is going to enter inside the cup-like structure. It is going to divide, re-divide to form a bundle or network of capillaries. Okay, there can be maximum 50 capillaries are there. So that bunch or bundle or knot of capillaries is known as glomerulus. Again, they are going to join, rejoin to form efferent arteriole. Okay, so this is a structure of Malpighian body that we have studied. If you see that this is a efferent arteriole, this is a efferent arteriole. Okay, the efferent arteriole has a larger diameter than efferent arteriole. Okay, efferent arteriole has larger diameter than efferent arteriole. So there is a difference in the diameter. Okay, there is a difference in the diameter. So diameter of the capillaries are still smaller in both the arteriole. Okay, such difference in the diameter where the blood flows with a greater pressure in the glomerulus. That means since the afferent has a bigger diameter, so blood flows rapidly inside the glomerulus, but it cannot leave rapidly because efferent arteriole has a smaller diameter. So blood is going to enter at the faster rate than it leaves. So because of that, there will be pressure created inside the glomerulus, and that pressure is known as glomerular hydrostatic pressure or capillary hydrostatic pressure. Okay, so there is a difference in the diameter. Efferent arteriole has larger diameter than efferent arteriole, so blood is going to enter into the glomerulus at faster rate than it leaves. So because of that, there is a pressure inside the glomerulus which is known as the glomerular hydrostatic pressure. Okay, or these are called as capillary hydrostatic pressure. Okay, it is one and the same. Now the innermost layer, okay, the innermost layer, that wall of the capillaries are extremely thin. Can you see this? This is the capillaries. Okay, they are extremely thin and porous. Okay, so the wall of the capillaries are extremely thin. Under the effect of high pressure, the wall become permeable to major component. That means whenever blood is flowing through that capillaries, okay, some of the molecule they can come out because of permeability. Hmm? So wall of the capillaries are extremely thin. Under the effect of high pressure, the wall become permeable to major component of the blood. Okay, so most of the component that can come out. Now, if you see that capillaries, these capillaries are in contact with the visceral layer of the Bowman capsule. And I hope you can remember that visceral layer is lined by the specialized cells called as podocyte. Can you see this is a podocyte? So this podocyte has a finger-like projections. Can you see these are the finger-like projection? So this is a modified squamous epithelium which is having the finger-like projection. So these are called as podocytes. So podocyte has a finger-like projection. So finger-like projection of one podocyte is going to interdigitate with the finger-like projection of adjacent podocyte, and then they are going to cause a filtration slit. And through that filtration slit, a blood get filtered. Okay, the blood get filter. So this filter blood is going to accumulate between the space which is present between outer layer and inner layer of the Bowman's capsule. So this is a space called as urinary space. Okay, this space is called as urinary space. That space is present between outer layer and inner layer. Okay, so outer is called as parietal, inner layer is called as what? Visceral layer. Okay, and the space is known as urinary space, or it is also known as capsular space. Okay, so after filtration, whatever that filtrate is formed, that filtrate is accumulated in the urinary space, and that filtrate is called as glomerular filtrate. Okay, that filtrate is called as glomerular filtrate. It is also known as deproteinized plasma. It is also known as deproteinized plasma because it has the same composition like plasma of the blood, except some cells, that RBC, WBC, platelets are not there. Okay, but the rest of the things are same like plasma. Even that glomerular filtrate do not have the protein, so it is called as deproteinized plasma. Okay, again I'm going to repeat, pay attention. Efferent arteriole has larger diameter than the efferent arteriole. So whatever blood comes in the glomerulus, it is under pressure because blood is going to enter at the faster rate than it leaves. Okay, so here the pressure is created. Okay, so under that pressure, okay, under that pressure, and the wall of the capillaries are extremely thin. They are highly porous. Okay, so because of the effect of the high pressure, the wall become permeable to major component of the blood except for blood cells and protein. Okay, so here the blood will get filtered. Okay, how that blood will get filtered? Across the filtration slits. Now what are the filtration slits? Filtration slits are formed by the podocyte. You can remember the visceral layer of Bowman capsule is lined by the specialized cells called as podocyte. So finger-like projection of one podocyte may interdigitate with a finger-like projection of another podocyte, and then they are going to act as a filtration slit through which that blood will get filtered, and after filtration that filtrate is going to accumulated into the urinary space. What is urinary space or capsular space? It is a space present between the parietal and visceral layer of Bowman's capsule. Okay, this is a space which is present between parietal and visceral layer of Bowman's capsule. Okay, so this is the first urine is formed, which is called as a primary urine, or which is also called as glomerular filtrate, or which is also called as a deproteinized plasma. It has the same composition like plasma, but it do not have the protein. So therefore it is called as deproteinized plasma. So you can see over here, that is ultrafiltration, separation of small molecule and ions from the large molecule of and cells in the blood. That is protein, large fat molecules are not filtered, but water, glucose, amino acid, salt, urea, uric acid, creatinine, drugs are filtered. That means that primary urine or deproteinized plasma or glomerular filtrate has all these substances: water, glucose, then amino acids, then salt, urea, uric acid, creatinine. So drugs, all these are get accumulated in glomerular filtrate. Okay, so it is also known as primary urine. It is also called as deproteinized plasma because it do not have the protein. Rest of the composition is same like plasma. Okay, so this is all about the process of urine formation. Okay, now you can see over here, this is again the part that is called as a Malpighian body. Okay, so you can see there are glomerulus. So glomerulus has a pressure. Why there is a pressure? Because of the difference in the diameter. Afferent arteriole has larger diameter than efferent arteriole. So there is a pressure inside the glomerulus, which is called as capillary hydrostatic pressure, or glomerular hydrostatic pressure, okay, which is around 55 mm of mercury column. Okay, so you can see over here, this is called as capillary hydrostatic pressure, which is approximately 55 mm of mercury column. That is a pressure due to the blood in the capillaries. Now that pressure is opposed by the two pressure, one from inside, another from outside. So inside the blood capillaries, there are proteins. So these proteins are going to oppose this capillary hydrostatic pressure. So that is known as osmotic pressure. Okay, so you can see there is a osmotic pressure. So osmotic pressure is because of the protein. Okay, blood is concentrated and will withdraw the water inward. Now what happens over here, the filtration of blood will take place. Okay, now here, the because of the filtration, now inside whatever blood is there, that blood become concentrated and it try to withdraw the water inward. So it is going to exert some pressure, and that pressure is called as osmotic pressure, which is around 30 mm of mercury column. So this capillary hydrostatic pressure is internally opposed by the osmotic pressure because of the protein. Okay, similarly you can see that here there is a formation of primary urine or glomerular filtrate or deproteinized plasma. It is also going to exert a pressure inward. Okay, so that pressure is known as filtrate hydrostatic pressure or capsular hydrostatic pressure. Okay, so the pressure due to the filtrate in the capsule, that is around 55 mm of mercury column. So this is 55 is opposed by 30 as well as 15. So if you take the cumulative, that is cumulative effect of the 30 and 50, so 30 and 15 plus 45. So 55 minus 45, that become 10 mm of mercury column, which is a total pressure inside the Bowman's capsule. So that is called as net filtration pressure. Can you see? Or effective filtration pressure. So effective filtration pressure is equal to capillary hydrostatic pressure minus osmotic pressure of the blood plus filtrate hydrostatic pressure. Okay, so what is capillary hydrostatic pressure? That is the pressure because of or difference in the diameter, or that pressure is because of the blood, because blood is going to enter at the faster rate. Okay, that is a pressure within the glomerulus. Okay, so this is around 55. So that is opposed by the osmotic pressure of the blood because of the protein, which is around 30 mm of mercury column, plus filtrate hydrostatic pressure, which is exerted by the glomerular filtrate or primary urine or deproteinized plasma, which is approximately 15. So 55 minus 30 plus 15, that become 10 mm of mercury column. So this is known as effective filtration pressure or net filtration pressure within the Bowman's capsule. Okay, and that is how that first urine is formed, that is called as glomerular filtrate or primary urine or deproteinized plasma. Okay, now always remember, both the kidneys are going to receive approximately 1200 ml of blood. Okay, both the kidneys are receive approximately 1200 ml of blood per minute. Okay, and out of that 1200 ml of blood, around 125 ml of glomerular filtrate is formed per minute. So the glomerular filtrate rate is 125 ml per minute. So you can see this is a glomerular filtrate rate. So 125 ml filtrate is formed per minute. That makes around 180 liters per day. In 4 minutes, entire blood passes through both the kidney, because 600 ml per kidney. So are approximately 1200 ml both the kidneys. So 1200 ml into 4, that become 4800 ml, which is approximately 4.8 liters. And in your body we have 5 liters of the blood. So filtrate is nothing but blood plasma minus protein. Therefore, it is also known as deproteinized plasma. Okay, so this is all about the first process called as ultrafiltration. Okay, so always remember, ultrafiltration is filtration under pressure. Okay, what is ultrafiltration? It is filtration under the pressure, and that is the first step of urine formation. And the first urine is formed is called as primary urine or it is also known as glomerular filtrate, or it is also known as deproteinized plasma. The next part that we are going to discuss over here, that is a selective reabsorption. That is the next step after ultrafiltration. Okay. Now, as we know that in the ultrafiltration, we have got a primary urine, okay, or which is also called as deproteinized plasma, or which is also known as glomerular filtrate. Now that glomerular filtrate contain all the vital substances. It also contain nutrient. Okay. Now we cannot lose the nutrient through the urine, so they have to reabsorb back. Okay. So the selective reabsorption takes place throughout the renal tubule. Okay, the selective reabsorption takes place throughout the renal tubule. Okay, so PCT is the major site of the reabsorption. So this is, you can see, this is called as proximal convoluted tubule. I hope you can remember that proximal convoluted tubule is lined by the cuboidal epithelial cells with microvilli. Okay, they have the cells with microvilli, which is going to increase the absorptive surface area, and they are involved in absorption of various substances, that is by a process known as reabsorption. So once that nutrient salt are reabsorbed, they are sent to the peritubular capillaries. Okay, so outward arrows that indicate the nutrient as well as salt or water is going to the peritubular capillaries. Okay, so there are two processes throughout the selective reabsorption. There are two processes are involved. One is called as passive absorption or passive transport, another is called as active transport. Okay, so what do you mean by passive transport? Passive transport, it is always from high cons or the passive transport is always from high concentration to low concentration across the concentration gradient. So when the molecule is going to move from high concentration to low concentration, so it is known as passive transport. So diffusion is an example of passive transport. Okay, active transport is always from against the concentration gradient, from low concentration to high concentration, and for that energy is required. Okay, so the selective reabsorption step that will occur throughout the renal tubule. Okay, so first you can see that primary urine is going to enter over here. In case of the proximal convoluted tubule, it will undergo the absorption of major substances. So major substances, that is amino acid, glucose, nutrient, salt, water, that is absorbed because they are lined by the cuboidal epithelial cells with microvilli. Okay, so these cells are going to increase the absorptive surface area. Now that urine is going to enter into the Henle's loop. Okay, as you know that descending limb of the loop of Henle, this is a descending limb. Okay, which is permeable to water, so there will be absorption of water from that region. Okay, and since there will be absorption of the water from this region, the urine become more concentrated. Now the further urine is going to pass into the next part called as ascending limb. In the ascending limb is permeable to salt, but it is impermeable to water. So here there will be absorption of salt will take place, and again urine become less concentrated. Okay. Now the urine is going to enter into the distal convoluted tubule where it can undergo absorption of water as well as salt, and finally urine is going to enter into the collecting duct. Okay. So you can see over here what are the steps that the selective reabsorption where the 99% of the glomerular filtrate is reabsorbed. So there are two steps or two processes are involved: passive transport and active transport. What is passive transport? It is always along the concentration gradient, that is from high concentration to low concentration. So example for or which substances are involved in passive transport, that is water and urea. Active transport, it is always against the concentration gradient from, that is, low concentration to high concentration. Sodium, potassium, amino acid, glucose, calcium, chlorine, they can be absorbed by active transport. Okay, now there are two type of substances. High threshold substances, they are completely reabsorbed in the blood. Okay, glucose, amino acid, we cannot lose that substances through urine. So they have to reabsorb back. So these are called as high threshold substances. Okay. Then there is one more substance called as low threshold substances. Low threshold substances, they are, absorbed in very small quantities. Urea and uric acid, they can be absorbed in small quantities. So they are called as low threshold substances. Okay. Then there is another substances are known as the no threshold substances. Okay. No threshold substances are those substances which are not at all absorbed. Okay. So these are called as what? No threshold substances. Example like creatinine, it is considered as a no or non-threshold substances which cannot be absorbed back. So basically there are three type of substances: high threshold, which are required for the body and they are completely reabsorbed. Then there is low threshold, they are reabsorbed in small quantities. Then there are no threshold, they are not reabsorbed at all. Example of the no threshold substances are creatinine. Okay, so this is the process called as selective reabsorption, where the 99% of the filtrate is reabsorbed. Now look at this table. Okay, now you can see there is a PCT. PCT is going to responsible both absorption, active absorption as well as passive absorption. You can see there is what absorption of water, urea, chlorine will take place. The glucose, amino acids, vitamins, major salt are absorbed in the PCT. In case of the descending limb, it is permeable to water, but it is impermeable to solute. So there will be absorption of water. So 5% of the water absorbed approximately is absorbed. Now here, due to increased medullary tissue osmotic pressure, right? Now here, the urine become hypertonic, more concentrated. Okay. Now ascending limb is impermeable to water, but it is permeable to salt. So there will be passive absorption of the salt. Okay. Active absorption of chlorine and potassium. So urine become hypotonic. DCT and collecting tubule, it is concerned with the passive reabsorption, and these are under the influence of the hormone called as antidiuretic hormone. So this antidiuretic hormone is responsible for more absorption of water from your urine, and that is going to make the urine more concentrated. Okay, so this is known as selective reabsorption. Okay, this is known as selective reabsorption. Selective reabsorption occur throughout the renal tubule. Coming to the next part is called as last part called as tubular secretion, or which is also called as augmentation. So finally, the filtrate will reach to the distal convoluted tubule via loop of Henle. Okay, I hope you can remember there is peritubular capillaries which is going to surround the DCT. Okay, so cells of the DCT and collecting tubules are actively absorb the waste like creatinine, iron, potassium, H plus ion from the peritubular capillaries and secrete into the lumen of DCT and PCT. Okay, यहाँ पे देखो, pay attention. Now you can see this first process. First process is called as ultrafiltration. Okay. The second process. Now, so what happens? Sometimes during the ultrafiltration, some substances are escape. They are going to escape and then they are going to enter into the peritubular capillaries. Okay. So the first step is called as ultrafiltration. Second is called as B is selective reabsorption. Okay. So during ultrafiltration, some substances are escape. They are going to enter into the peritubular capillaries. So they are collected and transfer back into the renal tubule. Okay. And that is called as tubular secretion. Okay, some substances which are escaped during ultrafiltration, they are going to enter into the peritubular capillaries. So they are collected and transferred back into the DCT and PCT, and that is called as tubular secretion, or it is called as augmentation. So which substances are normally that is collected and transferred back? Creatinine, iron like potassium. Okay, then there are H plus ion. So these can be absorbed from the peritubular capillaries and transfer back into the uh, renal tubule, and that process is known as tubular secretion, or it is also called as augmentation. Okay, so tubular secretion is the only mode of excretion in marine bonyfish and desert amphibian. Okay, that is the only mode of excretion in marine bony fish and desert amphibian. Okay, so this entire process is known as tubular secretion or augmentation. Now look at this. Substances are removed from the blood and secreted into the lumen of the PCT and DCT by active transport. Example is this. So creatinine, what is creatinine? It is a waste product which come from the creatinine phosphate. This is an energy molecule found in the skeleton. So once that energy is utilized, the creatinine become waste product. Okay, so it is found in skeletal muscle. So ammonia and creatinine is secreted in PCT, whereas H plus K plus ion that takes place in DCT is important to regulate the acidity of the blood. Sodium is reabsorbed, but excess is secreted. So antibiotics like penicillin, iodine containing compound called as iodoparacet, okay, that is used to check the contrast medium of your urine, that is going to use in renal function diagnosis. That means doctor will give you compound, that is iodine containing compound, that is called as iodoparacet, and they are going to check whether that compound is being excreted in urine. Okay, so that is how they are, the doctor will come to know whether your kidney is performing proper function or not. Okay, so this is all about the process of urine formation. So today we have discussed the first process called as ultrafiltration, second process is called as tubular secretion, and the third process is known, second process is selective reabsorption, and the third process, that is tubular secretion. So now you can see there is a difference between tubular reabsorption and tubular secretion. Tubular reabsorption is selective reabsorption. Okay, now you can see over here what is the first difference. This is the passage of selected substances from the ultrafiltrate into the blood. So whatever substances are being absorbed, they are transferred into the peritubular capillaries, that means into the blood. Here, the passage of selected substances from the blood to the ultrafiltrate. So some substance which escape during the ultrafiltration, they are collected and transferred back into the ultrafiltrate, that is into the urine. Now this is going to reduces the volume of the ultrafiltrate. This is going to increases the volume of the ultrafiltrate. Tubular secretion occurs by both passive as well as tubular reabsorption occur by both by passive as well as active transport. Whereas tubular secretion will occurs only by active transport. Tubular reabsorption, that is selective reabsorption, takes place throughout the renal tubule. This will exclusively occur in PCT and DCT. Okay, and these are the substances which are reabsorbed: glucose, amino acid, water, salt, little urea. The substances which are excreted are creatinine, H plus ion, okay, then uh, K plus ion, that is potassium ion, ammonia, etc. So that is the difference between tubular reabsorption or selective reabsorption and tubular secretion.